Hello all, I'm Achilles Pexaras, a biorefinery assistant scientist at the Integrated Biobank of Luxembourg, part of Luxembourg Institute of Health. And I would like to virtually welcome to my presentation, Learning from Cryobiology to Improve Biopreservation Quality in a Biobank. Biobanks are important infrastructures and they play an essential role in biomedical research. Biobanks connect various collection sites, clinics and hospitals with many research institutes worldwide. The role of a biobank is the creation and maintenance of an extensive repository of valuable biological samples, together with their associated data, that will be all used later on for subsequent research purposes. The life cycle of a biospecimen in a biobank has many stages, starting with the collection of the sample. The sample is transported at the premises of the biobank and where it will be processed and analyzed. The next step is the storage of the sample and its associated data, either for a short-term or for a long-term storage. If needed, there might be a further analysis and characterization of the biological material, and it will be finally distributed to the clients and researchers for downstream applications. If we put the life cycle in an horizontal sequence, we will see that there are many pre-analytical variables that can occur in this life cycle. There is a clear separation of the analytical variables that occur before the reception of the sample in the biobank, for example, during the sample collection and the transportation of the sample, and after the acquisition of the sample in the laboratory, mainly during the sample processing and the storage. It is really important to minimize these variables and to control them because they can change the molecular composition of the biological material and therefore affect the integrity of biospecimen. Let's have an example of a blood sample and its derivatives as the main source of biological material in a biobank. We can extract serum, plasma, and Buffy coat, which are stored at -80 degrees, and viable cells that are stored in liquid nitrogen, -196 degrees. Later on, from whole blood or Buffy coat, we can also extract nucleic acids, which are stored at -80 degrees. All these aforementioned biospecimen can be affected by some pre-analytical variables, for example, the time at room temperature and the centrifugation delays during the sample processing, as well as the interpersonal differences. For the sample characterization, the hemolysis, the freeze-thaw cycles, and the volume of the aliquot can have an impact as well. As far as the storage is concerned, the duration and the temperature, as well as the resilience of the containers, are important variables to consider. In order to control these variables and minimize the bias, in IBBL, here at the Integrated Biobank of Luxembourg, we offer automated processing and aliquoting of biospecimen. We perform automated transformation and derivation of biological material. We assess the fitness for purpose of our samples with various quality control tools. We biopreserve our samples based on cryobiology principles. And we assess the stability and the long-term functionality of the samples. So speaking today here among experts in cryobiology, I would like to focus on the last two topics, the biopreservation and the stability, talk about what we have learned and what we learned from this field, and what a biobank, how a biobank can implement this cryobiological background in its operations. So here there is a list, an example of functional assays that can be performed in many types of cells. And what we have learned is not only to use validated assays, but also to create a threshold range in which we will be based and accept our assays and thus assure and provide an homogeneous assessment. It is advised to use multiple levels of assessment, and in case that some laboratories are not able to validate their own techniques and methods internally, they can participate in external proficiency testing programs to check and verify their performance. Talking about biopreservation protocols, there are important and well-known parameters to take into consideration as listed here. In order to standardize these protocols, we learn to identify these critical parameters and temperatures and optimize each step of the thermal program, to understand the mode of action and the thermal behavior of the cryoprotectant, and also to use cryovials with good thermal conductivity. Another example about what biobanks learn from cryobiology and use it in everyday processes concerns the storage temperature. Different sample types have different optimal storage temperatures, and they can be biopreserved from a few hours up to many years. During the optimization and standardization of these protocols, we learn to choose a terminal storage temperature appropriate for each sample type, to put in place operational and risk management strategies, to monitor the temperature of the equipment, to use cryovials that have been tested and can withstand storage temperature and thermal freeze-thaw cycles, and also to consider the type of the plastic because it might affect downstream applications. Having established at IBBL these standard operating procedures, we still follow up the stability of our assays and the samples. Currently, we have 15 projects ongoing. Some of them are listed here. As an example, DNA and RNA extraction from frozen tissue stored in 2 different locations, -80 degrees and at liquid nitrogen, -196 degrees. The samples are processed and the derivatives are characterized for yield, purity, integrity, and applicability for downstream research. Taking this knowledge further, IBBL has developed a quality control tool. It's called findmyassay.com. It's an online, free and user-friendly tool that helps scientists to qualify their clinical samples and also to improve the reproducibility of their research. So in general, in order to achieve an optimal management of the biospecimen and the bioresources, we have established and developed standard operating procedures. We are working with validated assays and we follow the stability of our assays and our samples. IBBL is a certified and accredited institute by ISO. We calibrate and maintain our scientific equipment regularly. And last but not least, we are not only drivers, but also participants in the proficiency testing program. Plus, as I mentioned earlier, we have developed the findmyassay.com tool, which is a guide to qualify your clinical samples. So to conclude with, we have established a quality control check for every step of the biospecimen's life cycle. And that's how we can assure a high quality of samples, which will lead to better data and better science. Thank you for your attention. If you have any questions.
Thank you, Achilles. I'm sure everyone enjoyed this report